from Carnegie Mellon University. Today I'm going to talk about the efficient data-driven model learning for dynamical systems. This work is a collaboration with Iphigenia, Pranay, Paul Mark, and Professor Diana Maculescu. So for the simple biological dynamic systems, there can be several ways to do the analysis. So for simplicity, we can do some qualitative analysis, which could be a few descriptive languages. And we can also do the quantitative analysis, which could be the differential equations to model the details. However, when the system goes large, the quantitative analysis may not be available or may be very inefficient. Instead, there are several qualitative analysis which could be very efficient to model the system. Boolean function is one of those tools. So through the binarization, the system of the left can be converted into logic network which compose only 0 or 1 values to do the qualitative analysis. However, in reality, those detailed networks may not be available like always. So the lucky thing is that we have the experiment data or the kind of source data so that we can do some inference to reach the right side. Here's the outline of my talk. I will continue to introduce some background knowledge, including the big picture. I will also talk about the method, results, and finally, I will do the conclusion. So here is the big picture. So there are a lot of sources for the dynamic system data. They can come from the experiments, from the simulation, and from the people supplementary information if you want to do the massive collection. So through binarization, you can reach to the Boolean time series. There are several methods to do the binarization. It could be threshold-based. It could also be the k-means clustering and so on. So the Boolean variables can only be 0 or 1. So the meaning of them can vary from system to system. 0 can mean, for example, the low species concentration, while the high may be the high species concentration. There can also be other meanings. But through the Boolean time series, we need to do the inference to reach the Boolean function. So our contribution at this work is to propose an efficient inference engine to do this. So the right side shows the general data structure of the data we used. So there are n features with t+1 time steps. As a common process, the data usually are discretized in time domain. So the time intervals can vary from a few milliseconds to a few days, which is quite dependent on what you can get. So here's one of the concepts I want to introduce. It's called the Boolean model with a refractory delay, which means that the features you want to predict can be a function of the current time steps and a few previous time steps. So that's a general form. And the reason why refractory delay is important because in many biological systems, some features can have some delayed effect on others. For example, abundance or absence of a protein has to occur for some time before it can produce an observable effect. Canalizing feature is the second one I want to introduce. So that's the definition of the canalizing. actually it's a function if a variable, a value of the variable like 0 or 1 in Boolean can directly determine the full function, then we call the function is canalizing with this variable. And if it's more than one variable to have this feature, we call it a nested canalizing function. This is important because in many biological systems, the canalizing is very common. And it's also because this canalizing feature can lead to efficiency in the inference. Here's one example. So for all the features in this system, they are all canalized to some features. To start with our method, we will first go to problem formulation. So in Boolean algebra, conjunctive normal form is one of the common way to represent the Boolean function. So it's actually a product of sum. The feature, the property of this one is very clear. So if any of the sum is zero, we can see that the function will be zero. Only when all of the sum equals to 1, the function will be 1. So there's one obvious solution for this one. It's called Boolean satisfiability problem. And this problem has been proved to be NP complete, which means that we need to use super polynomial time to solve it. If the system is so large, this will be not efficient. So we propose a new greedy heuristic from positive and negative examples, which will be detailed in later. So our method can be divided into two phases. This is the first phase, we call it the data process. So we will iterate our algorithms from one feature to another. So to the illustration, we'll assume that there are 3 features in the example. And we also assume that the refractory delay is 0, which means that the function will be only dependent on the current time step value. A conjunctive normal form. If you wanted, it's not trivial. If you wanted, this is a conjunctive normal form. Yeah, maybe we can talk about it later. So we will use X3, the third feature, to do the illustration of the algorithm. So here's one example with 5 time steps with 3 features to do a very simple illustration. We need 2 containers to store the data. So for S0, it stores the instance that says feature as 0. S1 stores the instance that says feature as 1. So there are 2 containers out here, and we need to put the data or put the instance into the container. Let's say for the first row, that's the instance, and it results the X3 in the next step to 1. So we put it into S1 container. That's the representation of the instance here. X1 means the X1 variable equals 1. X2 prime means X2 variable equals 2. So that's a common way to represent in the Boolean algebra. So we can continue the process to fill the containers. Our target is to try to find a Boolean function that equals to 1 for all the instances in S0, and 0 for all instances in S0, and 1 for all instances in S1. We move on to the second stage. So we need to do the product of sum, so we need to infer each sum in the product. So for the each sum, we need to do the 3 stages. We need to pick the literals from S1 so that the sum in S1 all equal to 1. And we also need to check against with the S0 so that we need to make some instance in S0 to 0. If it's qualified, then we put it as the final one. So we can use this example to show the process. And the picking order is also determined by a indicator function. So this just means that the more often a literal shows in S1, the less often the literal shows in S0 will have the higher priority to be picked. So we can use this to say that with simple calculation, x1 prime will be picked as the first one to satisfy this 2 to 1. And we also need to pick S3 prime to satisfy the first instance to 1. And we check against the S0 with the sum of this, we can see that it will be 0 in this one. So since this is a very simple example, this sum will be the final solution for this. So for the simple analysis of the complexity, we can see that the algorithm is dependent on the number of features and also the number of the instance for the inference. So to be simple, it's only a n times omega in the complexity. Here the experiment setup, we will use 3 biological dynamic systems to illustrate our algorithm. And for comparison, we have 2 state-of-the-art methods. One is called best fit, which try to minimize the errors when inferring the variables. And the other is called full fit, which try to make the perfect inference for each variable with no error. So we use 2 key properties for evaluation. One is the runtime, one is the prediction error. So for the dynamic system, this one is simple compared to the other 2. This is the model for the differential process in TH cells. It includes 19 features, which they are all canalized. The second model we will introduce is the TLGL survival signaling network in lymphocyte leukemia. It includes 49 features, and that's the map. The third one is about the signaling network connections for the T cell differentiation, and it includes 47 features in it. So let's come to the results part. We first evaluate the error. So we use the average prediction percentage error, which the formula is shown here. And the meaning for this is just like the percentage of the mispredicted instance among all predictions. So the results are shown in this table. We can see that our method achieved perfect predictions in these 3 models, while the other 2 achieves some errors in the 2 large biological systems. And we continue to evaluate the runtime. So both the figure and the table show that our method, our proposed method, is more than like 1,000 times faster than the rest 2. One one thing to note is that our method is implemented in C++, but the other two was implemented. The other were implemented by a Berezovsky from Rice University. Was in Python. So we compare the runtime for the two implementation method. Usually Python and C++ is less than 10 times in the runtime. So we can claim that our method is 100 times faster than the other two. So we come to the conclusion. We explore the next canalizing features with respect to some the features in our inference algorithm. We also propose a polynomial time heuristics to infer the Boolean model. And the results show that our proposed algorithm runs more than 100 times faster than state-of-the-art algorithms while achieving a similar or better accuracy. Thanks for your time.
Very interesting work. So I have questions about your assumptions, actually. So you know the biological system is very, it's robust, but actually different biological features actually change across different scales. And you actually, if I understand correctly, you made two assumptions. The first assumption is that you just banalize the biological features, 1 or 0, right?